Hello, and welcome to this Nature Research custom media webcast titled Llama-derived Nanobodies in the Fight Against Parkinson's Disease. My name is Jayshan Karpan, and I will be your moderator. Today's webcast is produced by GenScript. We'll begin the webcast with a presentation from Dr. Wim Versées of Vrije University Brussels, and then end with a Q&A. To ask a question, just type it in where it says Ask a Question, and then press Submit at any point during the webcast, and we will answer them today. And now over to Wim.
Thank you for this introduction, and a warm welcome to everybody either joining live or listening later to this Nature webcast. And in this webcast, so during about the next 45 minutes, I will discuss how my team, together with a number of other groups, went from very fundamental studies in order to understand the mechanism of a protein involved in Parkinson's disease towards the development of so-called nanobodies against these proteins, which display interesting properties for use as novel therapeutic strategies to fight Parkinson's disease. And in order to do so, I will start with a short and therefore incomplete and somewhat simplified introduction on Parkinson's disease. So in fact, Parkinson's disease is the second most common neurological or neurodegenerative disease, only surpassed by Alzheimer's disease in the number of patients it affects. Indeed, worldwide, about 1 to 2% of the global population above the age of 65 is affected by this disease. And this results in a reported number of 6.6 million cases in 2015. However, due to the increasing age of the global population, this number is further expected to rise quite steeply and to reach about 14 million cases between 2030 and 2040. And although this disease is often referred to as a disease of old age, and indeed, as you can see here, the prevalence is clearly increasing with age, also younger people below the age of 45 can be affected by this disease, although that is clearly less common. So the most clear hallmark symptoms of this disease are all motoric symptoms, and they include tremor. So that is the typical shaking of the hands and the limbs one is quite familiar with. But apart from that, also slow and difficult movement and walking, postural instability and rigidity are typical motoric symptoms. And next to these motoric symptoms, there are also a number of non-motor symptoms, like, for example, loss of smell, problems with sleeping, gastrointestinal issues, and also depression, for example, and at later stage also dementia. So when we want to look a little bit deeper into what is actually happening inside the brain of people with Parkinson's disease, it is clear that especially the motor symptoms are due to a degeneration, so dying or malfunctioning of the dopamine-producing neurons in a part of the brain that is called the substantia nigra. And their dopamine is a neurotransmitter that is responsible for steering movement. And very often this degeneration of the dopaminergic neurons is associated with the occurrence of so-called Lewy bodies in the cytoplasm of these cells, where Lewy bodies are clusters of aggregated proteins, mainly consisting of the protein alpha-synuclein. Now, when we then even want to dig a little bit deeper into the mechanisms of Parkinson's disease and really understand the molecular pathways involved, it immediately becomes clear that these molecular pathways are very complex and still not completely understood. However, in general, Parkinson's disease can be classified in two large categories. The majority, about 90%, is the so-called idiopathic form of PD, which means that the exact underlying causes are not clearly defined and not completely known, although it is generally accepted that the risk to develop Parkinson's disease is a combination of environmental risk factors, like, for example, exposure to pesticides, and also genetic risk factors. And apart from that, about 10% of the cases are familial PD cases, which are monogenic, and mutations in about, well, a little bit over 22 genes, which are called PARC genes, are now clearly linked to the occurrence of PD. Now, very important to note is that, in fact, there is still no real cure for Parkinson's disease, and all the currently available therapies are only relieving the symptoms of the disease, but they are not really slowing down the progression or for sure not halting the progression of this disease. And one promising way to look for new um, um, ways, let's say, for finding new therapeutic strategies for Parkinson's disease is to really look at the mechanism of these PARC genes and to target these PARC genes and the proteins they encode. And one of these PARC proteins is the so-called Leucine-Rich Repeat Kinase 2, or LRRK2 in short, which will be the central topic of my talk today. You can see here the domain architecture of this protein. You can see it consists of many different domains. It's really a huge protein, and it is quite unique in the sense that it contains two catalytic activities in one single polypeptide chain. So the rock core domains here display GDPase activity, while the kinase domain, sorry, is a serine-threonine protein kinase that both autophosphorylates LRRK2 itself, so phosphorylates serines and threonines within the protein, and also phosphorylate downstream effector proteins. And mutations in the gene coding for LRRK2 are among the most common causes of the familial form of Parkinson's disease. And these mutations are now also linked to certain other inflammatory diseases, such as Crohn's disease. It is very important and interesting to note that all these mutations are dominant gain of function mutations in the sense that they increase the kinase activity of the protein and decrease the GDPase activity. And it's important to bear that in mind for later during my talk. Now, lately, overactivation of LRRK2, for example, overexpression of LRRK2, has also been associated with the much more common idiopathic form of Parkinson's disease. And that, of course, all these things combined, makes LRRK2 a very attractive target for drug development. And so a few years ago, it was shown by the groups of Dario Alessi and Susan Pfeffer that one of the major class of substrates for the protein kinase activity of LRRK2 are a subgroup of so-called RAP GDPases, which are small G proteins and which get phosphorylated on their switch 2 loop, although for sure also other proteins have been suggested as being substrates for the protein kinase activity of LRRK2. And then via these phosphorylated RAP proteins or via alternative routes, LRRK2 plays a role in important cellular processes such as vesicular trafficking, autophagy, and lysosomal function. And considering the central role of LRRK2 in PD, in Parkinson's disease, of course, it has been, or many different strategies have been suggested to target this protein therapeutically. But let's say the main group of strategy or the most important strategy for now has still been to design ATP competitive kinase inhibitors, so molecules that bind in the kinase ATP binding pocket and thereby displacing ATP and in this way inhibiting the kinase activity. But as you can see here, also different other strategies to target LRRK2 have been proposed. Yeah, and it is there in these type of strategies that we will come in with this story. However, before focusing on the targeting of LRRK2, let's first zoom out a little bit and look at LRRK2 itself. So in fact, LRRK2 belongs to a much larger family, which are called the Rocco protein families, which are conserved in all domains of life. So they occur in certain bacteria, archaea, and also in higher eukaryotes. And a kind of the central motive uniting all these proteins is the occurrence of these rock and core domains here, which always occur together, and where rock stands for RAS of complex proteins, and this is a G protein, so this domain bears the GDPase activity, and it is always followed by this core domain, which then also just stands for C-terminal of rock, and so this should really be considered as a kind of tandem domain. And based on the domain arrangement of the remainder of the protein, these rockoproteins have then been divided in three groups. Group 1 are the simplest members, with just a leucine-rich repeat domain in front of the rock core domains. The group 2 rockoproteins, to which also LRRK2 belongs, as you can see here, is more diverse in terms of domain arrangement, but they contain, in addition to the LRR on the N-terminus of the rock core, always a protein kinase domain at the C-terminal end of the rock core, and the group 2 proteins, they have the kinase domain in front of the rock core. Now, in order to study the mechanism of LRRK2, we in fact started by studying a much simpler model system from the group 1 proteins. And in fact, for that, we used the protein from a bacterium Chlorobium tepidum, which you can see here. So that just consists of the LRR, the rock, and the core. Is as such a much simpler model system than LRRK2 itself, does not contain the kinase domain, and that's of course an important point, but it turned out to be an excellent model to study the function of this highly conserved rock core domain module, which is central to all the rocko proteins. And so the first thing, in fact, we did was we were able to crystallize this protein and then to solve the X-ray crystal structure of this protein, which is displayed here. And that was, in fact, the very first structural information, or at least the very first experimental structure of any LRR rock core arrangement. And so it shows here the protein as a homodimer with two subunits, the two LRR domains in yellow, the core domain in blue, and the rock domain in red. And in fact, a few years after we were able to solve this crystal structure, and thanks to the revolution in cryo-EM technologies, it has also proven to be possible to solve structures not by our group, but by other groups of the human LRRK2. First in 2020 in the Leschinger group, a structure was solved and published of the C-terminal part of LRRK2, so containing all the catalytic domains. And then very recently in 2021, the group of Yisun and co-workers were able to solve the cryo-EM structure of full-length LRRK2. But even before these two structures were available, we continued using our Chlorobium tepidum rockoprotein, so the bacterial LRRK2 ortholog, to dig deeper into the mechanism of this protein, mechanism of regulation and the mechanism by or the mechanism of its functioning. And a first important observation we made was done by small angle X-ray scattering, which in fact showed, if you look here, that this protein undergoes quite large conformational changes depending on whether it is in the nucleotide free form, bound to GDP, or bound to GDP. And in this respect, it is important to note that the crystal structure that we solved, and which shows the protein as a homodimer, was bound in its nucleotide free form, so without any GDP or GTP bound. And then using multi-angle light scattering, we in fact found that while the protein, as we saw it in our crystal structure, was a dimer in the nucleotide free state and also in the GDP bound states, we found that it was a monomer when bound to GTP. And that was also confirmed using native mass spectrometry, where again, we mainly see here peaks corresponding to the dimer in the nucleotide free form and the GDP bound form, while when bound to a stable GTP analog, so GPP NHP is just a stable analog, a non-hydrolyzable analog of GTP, we see a distribution of dimers and monomers. Moreover, in negative stain electron microscopy, where we use way lower protein concentrations than in the other methods, we again see the protein in a dimeric form in the nucleotide free state. So you see these X-shaped particles, which are dimers, while we see nearly only monomers in the GTP bound state. So these more elongated worm-like particles are the monomers. But at those low concentrations, we see a mixture of dimers and monomers when bound to GDP. And all that together led us to conclude and to come to a model which I think is quite interesting and to my knowledge is also quite unique, where the protein cycles between dimers and monomers depending on the nucleotide that is bound to the protein. So when we go through this cycle, you see that the protein is a dimer in the nucleotide-free state, as we also observed in the crystal structure. Upon GTP binding, it monomerizes, and after GTP hydrolysis, it redimerizes again, first through this more labile dimer in the GDP-bound state, and then a stable dimer in the nucleotide-free state. Moreover, we found that this cycle is relevant for the disease mechanism, since we showed that mutations that we made in the bacterial protein and which mimic the mutations that cause Parkinson's disease in the human protein, at first, well, they also decrease the GDPase activity, like also mutations, Parkinson's disease mutations in LRRK2 do, as I showed you here. But moreover, we could show that these mutations slow down the GDP hydrolysis rate, as you can see here. So this is the wild type protein, and this is the mutant. And so you clearly see that the GDP hydrolysis goes down. And this is caused by these mutations blocking the protein in the dimeric state, or at least hampering the transition from the dimer to the monomer. As a consequence, the protein cannot cycle that efficiently anymore through this dimer monomer cycle, and the GTP hydrolysis is being slowed down. And this, of course, immediately triggered the question to us, can we affect these equilibria, and can we kind of counter the effect of these mutations by interfering with this monomer dimer equilibrium and thereby, let's say, nullifying the effect of this mutation. So we want to find molecules that can change this monomer dimer equilibrium. And one class of molecules that are known to be exquisite tools to really bind proteins in a conformation specific way and would have the ability to change this equilibrium are the so-called nanobodies from the title of my talk. So what are these nanobodies? Well, they are in fact fragments of antibodies which occur in animals belonging to the family of the camels. So this family not only contains camels, but also dromedary, llama, guanacos, alpacas, and vicuñas. And just like any other animal, and just like you and me, they contain conventional antibodies, which, as you might know, consist of 2 heavy chains, here in yellow and orange, and 2 light chains in these greenish colors. However, these animals contain an additional group of antibodies which lack the light chains and are therefore called heavy chain only antibodies. And this means that while conventional antibodies use 6 loops to bind their antigens, so 3 loops are here on the tip of this VH domain and 3 loops are present on the tip of the adjacent VL domain. However, the heavy chain only antibodies only use 1 domain to bind to their antigens, to their target proteins. And so they only have 3 hypervariable loops, also sometimes called CDR loops, at their disposal to bind these antibodies, to bind their target proteins, sorry. And therefore, the loops of this VHH domain are on average, let's say, somewhat longer than the corresponding loops over here, and especially this 3rd loop in red is on average longer than the corresponding loop in the VH and VL domains over there. Now, this means, since these heavy chain antibodies use only 1 single domain for binding to their antigens, that this domain can be cloned as a standalone protein, which remains or contains all the binding capacities of the parental antibody, and which is then called a nanobody. So when we look at the properties of these nanobodies, well, since they are single domain, they are extremely easy to clone and to engineer. They can be quite easily recombinantly expressed, for example, also in bacterial expression systems, and they can be easily purified. They are small, about 15 kilodalton, which is 10 times smaller than a conventional antibody, extremely stable and robust, and they can be expressed in the cytoplasm of eukaryotic cells, for example. And this means, of course, that these antibodies have found multiple applications as research tools, but also in diagnostics for imaging and more also in therapy. And a last property of these nanobodies that I would like to emphasize, because that's important for the applications we have in mind, is that due to these longer loops that these nanobodies have compared to classical antibodies, these loops have the tendency to really protrude and bind into cavities of their target proteins, as you can see here for a nanobody binding with its CDR3 loop into the active site of lysozyme. And that makes that these nanobodies are very suited to bind proteins in a conformation specific way. So if there are different conformations of the protein, the nanobodies might recognize only one of them, and so to also change the equilibria between conformations. Okay, so one can then wonder, how can we obtain nanobodies against a certain antigen, against a certain protein? And in fact, for that, there are different methods and strategies. Some of them are described in these papers here. So we can start from different types of libraries, synthetic libraries or immune libraries. There are different ways to present and select the nanobodies, like yeast display, ribosome display, or phage display. But we still get the best results by really going through an immunization of a llama, in our case, with the protein, with our target protein. After a few weeks of immunization, we take some blood, not very much, of the llama, and we clone the nanobody repertoire into a phagemid library, which then allows to express these nanobodies on the tip of a filamentous phage. And this then allows us to perform a few rounds of phage display panning to select the correct nanobody, so the nanobodies that bind to our protein of interest. Then we always go through an ELISA validation step to verify indeed and confirm that our nanobodies are binding to our target protein, and then these nanobodies can be used for a further thorough characterization. And so we then wondered, indeed, as I already told you, whether it would be possible to make nanobodies that bind to our bacterial LARC2 protein and would change this monomer-dimer equilibrium. So bind the protein in a conformation-specific way. So we immunize the llama with either the full-length protein or just a ROCK-core domain construct, and this in the presence of a stable GTP analog to drive the protein to its monomeric state. And via the methods that I just explained, it was then possible to find a range of nanobodies, two of them which display interesting properties, which we call nanobody ROCK1, which binds somewhere within the ROCK-core, and nanobody ROCK2, which binds somewhere within the LRR. And as we were actually aiming for, these nanobodies indeed bind our ROCK protein in a conformation-specific way, or in a nucleotide-specific way, in the sense that they have always a higher affinity for the monomeric forms than for the dimeric forms. And this is most pronounced for nanobody ROCK1, which does not bind at all or only with very low affinity to the stable dimeric nucleotide-free form of the protein, while it gradually binds better and better if the protein becomes more and more monomeric, as you can see here. And moreover, and this is quite interesting, we also saw that these nanobody, and especially this one here, can significantly increase the GDPase activity of our chlorobium sapidum ROCK protein, and in that way also counteract the effect of Parkinson's disease mutation. So as I told you, and as you can see here, the Parkinson's disease mutations decrease the GDPase activity. So you see here in gray the normal wild type protein or the GDPase activity of the wild type protein, which is significantly decreased in the mutant. But when we then add in blue the nanobody to this mutant, the activity, the GDPase activity, is completely restored and is even higher than in the wild type protein. And moreover, we in fact show that this is due to the fact that our nanobody indeed shifts the equilibrium from the protein, from the dimeric state to the monomeric state. So you can see that here on the left. So this is again native mass spectrometry. Here for the wild type CT ROCK protein that, as I showed you, in, let's say, or at least at the concentrations used in native mass spectrometry, was mainly dimeric in the GDP bound form. But when we then add our nanobody to this protein, you can see that it is shifted towards the monomer. And moreover, if we look at the mutant, so the Parkinson's disease mutation, which in contrast to the wild type protein remains dimeric when bound to GTP. So the wild type there is monomeric. Here it is mainly dimeric. But when we add the nanobody, it also becomes nearly completely monomeric. So again, there, our nanobody is able to revert the effect of the Parkinson's disease mutation. So if we look at that on a scheme, in fact, the nanobody completely counteracts the effect of the mutant. So while the mutant shifts the monomer-dimer equilibrium towards the dimer, our nanobody shifts it towards the monomer and thereby also increases the GDPase activity of this mutant and kind of reverts the Parkinson's disease effect of this mutation. And moreover, we were also able to show, but I will not dwell too much on that, that our nanobodies do that in an allosteric way. So not just by binding on the dimer interface, which would permanently block the protein in a monomeric state, but by more subtly changing this equilibrium. And at least we think that this is quite interesting and might open up new routes for thinking about therapeutic strategies to target Parkinson's disease mutations in LRRK2. But of course, I kind of hear you thinking even through the internet collection, well, that's all very fine, but this is all done with a bacterial ortholog of LRRK2. So what about human LRRK2? Well, of course, we were posing ourselves the same question. So we now wanted to see whether it was also possible to generate nanobodies that can affect the activity of human LRRK2. And now, since we already focused before on the GDPase activity, we wanted to focus on the kinase activity. So we purified the full-length protein and we generated or received a number of shorter domain constructs of this protein, as you can see here. And with all these constructs in hand, we followed now 3 immunization strategies where we immunized 3 different llamas with different proteins or proteins in different states. So one with the ROCK-core domain here in the presence of GPP-NHP, so a stable GTP analog, and 2 other llamas we immunized with full-length protein either in the presence of GDP or GTP in order to find conformation-specific nanobodies. And then after 1 to 3 rounds of phage display panning, as I explained you a few slides ago, we were able to retrieve 168 different sequences of nanobodies, which is a huge number. And therefore, we decided to only purify 42 of these. And then we used our ELISA approach to verify whether these nanobodies were binding on LRRK2. But in this case, we didn't only coat our ELISA plates with LRRK2, but also with a number of the domain constructs that I just showed you in order to see where on which of these domains our nanobodies are binding. So we did that with the ROCK-core, the ROCK, the C-terminal part of the core domain, which we call core B, the kinase WD40 domain, and in fact also the armadillo domain, but that is not shown on this graph here. And the results of this effort you can see here in this scheme. And in fact, we could see that from the 42 nanobodies that we tested, 40 in fact do bind on LRRK2. So 2 were probably false positives or had a very low affinity. And of these 40, the majority, 18, were binding in or within, somewhere within at least this WD40 kinase domain part. 5 were binding on the ROCK, 7 on the C-terminal part of core, 5 on the armadillo domain, and 5 others we call full-length LRRK2 binders, like for example, nanobody 38 here, because they bind on full-length LRRK2, the black bar, but do not bind on any of our tested individual domain constructs. And I will come back to that in a few moments. So since we now have a variety of nanobodies binding a variety of domains, we then came to our central question, and that is whether our nanobodies can indeed affect the kinase activity of LRRK2. And for that, we used first a cellular assay where we co-transfected human cells with the G2019S mutant of LRRK2. This is the most commonly found Parkinson's disease mutant in LRRK2, which significantly increases the kinase activity. And we co-transfected this in the cell with our nanobodies. So one by one, the nanobodies that we wanted to test. And we looked then at 2 activities, autophosphorylation at this position and phosphorylation of RAP10. And this we did using a Western blot approach, where we were using phosphospecific antibodies against this position, or specifically recognizing the phosphorylated RAP10. And you can see the results of such a representative Western blot in this picture here, and I will slowly walk you through it. So in the upper two rows, you can see the levels of phosphorylated, so autophosphorylated LRRK2 at two different development times of the Western blot. The next three rows show you the phosphorylated RAP10 levels, again at different times of development, and the lower three rows here on the gels are just expression controls to see whether all our proteins are indeed present in the cells. And in the blue boxes here, you see the negative control. So those are cells just transfected with the G2019S mutant of LRRK2 without any nanobodies. So these are giving kind of the reference levels of the phosphorylated proteins in the cells without the nanobodies. So you have to compare the intensity of the other bands with these intensities here. And then you can see that we have different activity profiles of the nanobodies. Of course, you have neutral nanobodies, which means that they bind on LRRK2 but do not affect the activity of LRRK2. For example, nanobody 9, 10, 13, when you compare the intensity of the bands in the blue box and here, you see that they do not affect these levels. Then you have nanobodies, like the one here boxed in red, that clearly activate the kinase activity, since the bands become more intense. Then, and that is what we were looking for, in fact, we have nanobodies like the one in dark green that clearly inhibit the kinase activity, both autophosphorylation and RAP10 phosphorylation. And finally, and I think very interestingly, we also find nanobodies of the light green type here, which do not affect autophosphorylation, but specifically inhibit RAP10 phosphorylation, as you can see here. And so with this then in hand, we now selected 10 nanobodies coming, let's say, from these different activity groups, neutral, activating, inhibiting RAP10, and inhibiting all activities, for a further thorough biochemical analysis. And first of all, and I will go quick on that in interest of time, we wanted to confirm and refine the domain mapping that we did using ELISA, so to see where the nanobodies are binding. But now we used a cross-linking mass spectrometry approach, where we cross-linked the nanobodies with LRRK2 and then saw which regions of LRRK2 are cross-linked with the nanobody to see approximately where these nanobodies are binding. And you can see the results of this effort here, with the solid lines representing cross-links here with LRRK2 and these representing our nanobodies. And I will not dwell in all the details, but basically we could largely confirm the results coming from the ELISA. And we can also see that these so-called full-length LRRK2 binders that were only binding on full-length LRRK2 but not on the individual domains, and nanobody 38, 36, and 17 are examples of those, that they indeed form cross-links with domains at really the N-terminal side and C-terminal side of the protein. So they probably really bind on the interface of different domains and therefore need the full-length protein in order to bind. Next, we wanted to see how strong our nanobodies are binding to LRRK2, so the binding affinity. For that, we use two different complementary techniques that I will not completely explain here again in interest of time. That's microscale thermophoresis and biolayer interferometry. But basically what we found is that all nanobodies bind with high affinity to LRRK2 with KD values, so equilibrium dissociation constants, in the low, or well, let's say single-digit to double-digit nanomolar range. So now we know that we have nanobodies that bind strongly to LRRK2 and that modify the activity of LRRK2 in cells. So we now wanted to see whether these nanobodies can also affect LRRK2 kinase activity in vitro, making use of purified LRRK2, purified nanobody, and purified substrates. Again, we use two different approaches here, two different assays. One where we looked at phosphorylation of a small peptide substrate as a model substrate, and one where we looked at the genuine substrate RAP8A. And you can see the reference levels here, where we just add no nanobody and where we just look at the basal activity of LRRK2. And then reassuringly, you can see that the red type of nanobodies that we're activating in cells also activate here in vitro. Of the 4 dark green nanobodies that we tested, so that means that they inhibited both autophosphorylation and RAP phosphorylation in cells, we see that 3 correspondingly also inhibit peptide phosphorylation and RAP phosphorylation in vitro. There's one outlier, nanobody 42, which clearly inhibited RAP phosphorylation in cells, but does not seem to do so in vitro. And we have no clue yet what is going on there. Maybe the nanobody, by binding to LRRK2, just changes the localization of LRRK2 in the cell so that in the cell it cannot reach RAP10 anymore. But that we don't know, this is just speculation. And then finally, and I think again very interestingly, we find that these light green nanobodies, which only inhibited RAP phosphorylation in cells and not autophosphorylation, correspondingly also only significantly inhibit RAP phosphorylation in vitro and not so much peptide phosphorylation. So all this summarized, we end up with 5 different groups of activities. Group 1, inhibit all tested kinase activities of LRRK2. Group 2, only inhibit RAP phosphorylation, both in cells and in vitro. Group 3, stimulate the kinase activity. Group 4, although I may be a little bit over-enthusiastic calling this a group since we only have one representative of those, only inhibit kinase activity in cells, and group 5 are just neutral, bind to LRRK2, but do not change the kinase activity. So now that we know at a single concentration that we used here of nanobodies that some of them inhibit kinase activity, we next wanted to see whether these group 1 nanobodies, so we now specifically focused on these, also inhibit LRRK2 kinase activity in a dose-dependent way. So we gradually used more and more nanobodies and looked at the LRRK2 kinase activity using again the peptide as a substrate, and basically we can see that all 3 nanobodies indeed show nice dose response curves with IC50, so values where half of the protein of the LRRK2 is inhibited, in the, let's say, low micromolar range, a little bit worse for this one here. And I admit that there is still room for improvement to increase the potency of these nanobodies. So since we now know that we have nanobodies that inhibit LRRK2 kinase activity, it's maybe worthwhile to look at this scheme here. In fact, in literature, kinase inhibitors are classically divided in different types, with type 1 and type 2 being inhibitors of the ATP competitive type. So that means that the inhibitors are binding in the same binding pocket as ATP is doing. And so in fact, ATP and the inhibitor compete for each other. So the inhibitor kicks out ATP and in that way inhibits the kinase activity. However, in the type 3 and type 4 kinase inhibitors, these inhibitors bind in a different pocket and so inhibit the kinase activity in a non-ATP competitive way. So they bind in a so-called allosteric pocket away from the ATP binding pocket. And it's important to note that nearly all currently available specific LRRK2 kinase inhibitors are of this type 1 group of inhibitors. So they are ATP competitive. So we were now wondering whether our group 1 nanobodies, so the one that inhibit all kinase activities, are ATP competitive or non-ATP competitive. And why am I only focusing on the group 1? Well, the group 2 only, of course, inhibit RAP phosphorylation and not peptide and autophosphorylation. So by definition, it means that these nanobodies cannot compete with ATP, because if they would kick out ATP of the active site, they would completely inhibit the kinase activity. So since we know that the group 2 are by definition ATP allosteric inhibitors, we now focused on our group 1 nanobodies. And to see whether they are ATP competitive or not, we first used a thorough kinetic analysis. Again, in interest of time, I will not have the time to really go in all the details, but basically we determined full Michaelis-Menten curves, so meaning that we measured kinase, the rate of kinase activity in function of the ATP concentration. So these are these individual curves that you see here. And for each nanobody, we did that at different nanobody concentrations represented by the different colors. And then we performed some linearizations, and we did in silico fitting, so computer fitting of this data, which showed us that all 3 nanobodies act as so-called mixed type inhibitors or non-competitive inhibitors, so meaning that all 3 nanobodies do not compete with ATP and so act as ATP allosteric inhibitors, different from the commonly used or available LRRK2 inhibitors that are available now. And maybe the easiest way to appreciate that they do not compete with ATP is to look here at these curves, and you can see that the nanobodies decrease the maximum velocity. So this is at low nanobody concentration, this is at high nanobody concentration. So they decrease the maximal velocity, meaning that even at very high ATP concentrations, the nanobodies still inhibit, so ATP cannot outcompete the nanobodies. And to further confirm this, we used a second approach, a competition ELISA titration, so where we coated an ELISA plate with LRRK2, either in the absence or presence of MLI2. And MLI2 is a very tightly binding type 1 kinase inhibitor of LRRK2. So it really binds in the ATP binding pocket. And then we titrate this with increasing amount of nanobodies, and we look at the binding of these nanobodies. And of course, if the nanobody would bind in the same pocket as MLI2, so in the ATP binding pocket, this binding would be hindered compared to this binding here. And you can see that by comparing here the green and the blue curves. The other 2 curves are just controls. And as you can see, the green and the blue curve, so either in absence or presence of MLI2, completely superpose. So that means that MLI2 is not able to compete out the nanobody, so that the nanobody is binding in another pocket than MLI2 is doing, and so differently from the available kinase inhibitors. And this is quite important and quite significant because it was quite recently, again in 2020 to 2021, shown in the groups of Leschinger and Villa in San Diego that binding of a type 1 kinase inhibitor, or binding of all tested type 1 kinase inhibitors to LRRK2, cause a conformational change in LRRK2, and this subsequently causes a relocalization of LRRK2 from the cytoplasm to microtubules, where it forms these kind of filamentous structures. And these filamentous structures, in turn, hinder the transport of cargo along the microtubules by motor proteins. And this might or might not be linked to adverse effects, so negative effects that are observed in, for example, lungs and kidney of animals that are treated for a long time with high doses of kinase type 1 inhibitors. So we thought it was important to check whether our nanobodies also cause this relocalization, this unwanted relocalization of LRRK2 to the microtubules. To do that, we co-transfected again our human cells with LRRK2 now linked to mCherry, which is a red fluorescent protein, and our nanobodies to GFP, which is a green fluorescent protein. And we then saw whether our nanobodies cause LRRK2, so the red fluorescence, to relocalize to microtubules. And in contrast to MLI2, we found that none of our nanobodies seem to have that effect. But of course, it's much harder to show that something is not happening than to show that something is happening. And so therefore, we also used another approach where we now again, as you can see here, co-transfected the cells with M-scarlet LRRK2, so the red fluorescence, and GFP nanobodies, the green fluorescence, and here it's the superposition of the two, or the merger of the two. But now we pre-treated these cells with MLI2. And then indeed, if you treat the cells with MLI2, you see in the controls, so where we didn't add any nanobody, but just GFP, or where we transfected with an irrelevant nanobody, that indeed these filamentous structures along the microtubules are being formed. However, with a subset of our nanobodies now, so 6 out of the 10 tested nanobodies, clearly revert this effect of MLI2 and inhibit this effect of MLI2, meaning that these nanobodies really bind in another way than MLI2 and even can or are able to block LRRK2 in a conformation that is different from the MLI2 conformation. And we now would really like to see whether that also has consequences in terms of adverse side effects of, or any adverse side effects of our nanobodies. But these are still experiments that are going. So we now clearly need to go with these nanobodies to more relevant neuron-like cells and also some relevant model organisms to further test them. But that is ongoing, and who knows, maybe that could be the topic of a later Nature webcast. But so at this point, I would slowly like to finish up and to wrap up by just giving some conclusions of the work. Maybe first some conclusions on the LRRK2 related work, so the second part of my talk. So there we showed, because that are the most, let's say, medically relevant findings, we could show that the nanobodies, or that we found nanobodies binding with high affinity to different domains of LRRK2. And these nanobodies inhibit LRRK2 kinase activity and using different mechanisms. So inhibiting different aspects of the LRRK2 kinase activity. And they do that in a non-ATP competitive way. And as a result, and different from the commonly available type 1 inhibitors, they do not induce LRRK2 relocalization. And we are convinced that these nanobodies will form great tools to be used in further research, but they might also have more direct applications in diagnostics and for the development of future therapeutics, but much more testing is needed for these latter approaches. And then as more general take-home messages, I would like to convey the message that, in fact, simple model systems, even bacterial model systems, can provide very valuable mechanistic insights that can be extrapolated sometimes also to the human proteins. And such type of basic science type research to gain fundamental mechanistic insights in the target proteins, in proteins involved in diseases, are extremely important and can lead to new out-of-the-box therapeutic strategies. And so finally, of course, nanobodies form excellent tools, as I've shown you, to allosterically modulate the activities of proteins and enzymes in general, and disease-related proteins and enzymes in particular. So to finish up, I would just like to thank a few people. Of course, my group. So this is my current group who performed, well, without whom I would, of course, have nothing to tell you today. Also, some former lab members were involved in this work. And I would also like to emphasize that everything I've been telling you today was in fact a result of a very close collaboration with these three groups here from the start. So we collaborated with the group of Arjan Kortold from the University of Groningen, Johannes Glöckner at the DZNE in Tübingen, and Frits Herberg at the University of Kassel. And also many other groups contributed in several different ways to the results I've been presenting. And I would also, of course, like to thank my host institutes and funding bodies for funding this work. And finally, I would like to thank you for tuning in, for your very kind attention. And I'm in fact very much looking forward to address some of your questions.
Excellent. Thank you for your presentation, Wim. It's now time for the Q&A. To ask Wim a question, all you need to do is type it in where it says questions and then press submit. So Wim, our first question asks, what potential ways are there to deliver nanobodies into the brain?
Yes, that's of course an extremely good question and well, not an easy one to address, let's say. But of course, we have been thinking about that. So there are two ways, in fact, to, let's say, try to valorize these nanobodies that we have now. That's either using the nanobodies as such directly as a kind of therapeutic compound, or to mimic their effects once we have structural information of where and how they bind and how they affect the activity, by targeting the same pocket with small molecules, for example, that would be brain penetrant and could cross the blood-brain barrier. But obviously, except for some exceptions of very positively charged nanobodies, they do not cross the blood-brain barrier. And so then we need certain ways to deliver these nanobodies to the brain, as indeed correctly pointed out. And that is still not an easy task to achieve. So, of course, for example, in model organisms, we could think of intraparenchymal injections directly into the brain, but that is not so obvious and very invasive for patients, of course. But nowadays, more and more viral delivery systems, so gene therapy approaches, are being tested, valorized for delivery of these of proteins, and especially nanobodies, which are quite small to the brain. So, for example, AAV type vectors, so adeno-associated viruses, are now being developed that can cross the blood-brain barrier. And I think at the moment, about 50 or so clinical trials have been starting up with these gene therapy approaches, so AAVs, for delivery to the brain. And the first one is on the market now. That is Zolgensma, I think, is the commercial name. So that's the drug that is now on the market against SMA, so spinal muscular atrophy. But of course, that is one of the biggest hurdles of directly using nanobodies for therapeutic approaches for the CNS.
Excellent. Thanks, Wim. And has anyone tried making nanobodies to alpha-synuclein aggregates?
I'm not so sure that there are against alpha-synuclein aggregates, but for sure, and that's not our work, although some people in our department have been involved in that study, there are nanobodies against alpha-synuclein that have even been tested in mice and were used in a kind of PROTAC strategy, so where you fuse the nanobodies, and that's one of the advantages, you can easily engineer them, so they were fused to peptides that then target these nanobodies and anything that is bound to the nanobody to the proteasome for degradation. And in that way, they could quite efficiently lower the burden of aggregated alpha-synuclein in the brain of these model mice. So just by degrading the alpha-synuclein via the proteasome.
Excellent. Thanks, Wim. Our next question asks, what is known about why and when camelids make both traditional and short VAH antibodies? Is there a trigger, an environmental or target type trigger to bias towards which type of VAH gets produced?
Not as far as I know, but I wouldn't be able to answer that with 100% certainty. As far as I know, when we immunize a llama with any type of proteins, both type of antibodies are being produced against them. But so we immediately clone the VHH repertoire from the white blood cells. So we kind of get rid of all the classical antibodies. But as far as I know, both are being produced. And then, of course, the million-dollar question in a way is, why did only llamas develop these heavy chain antibodies, but also sharks for some reason have a type of heavy chain antibodies somewhat different. And that is, as far as I know, not really known. They don't protect as well against bacterial infections and for example, fungal infections. Maybe they are as good against viral infections. So somehow in the evolution, there must have been a clear advantage for camelids to produce these heavy chain antibodies, but as far as I know, it's not really known why evolution chose some way to develop these heavy chain antibodies in camelids.
Excellent.
But it's an interesting question in a way.
Yeah, absolutely, and I expect more research can be done on those points. Our next question asks, do you have any idea how the NBs would affect LRRK2 variants associated with Crohn's disease?
So you have variants that indeed, so mutations that lead to Crohn's disease, and also these variants in a way act in the same way as the PD mutants. So they overactivate the kinase activity. So also these mutations lead to an increase in kinase activity. So I'm pretty sure that our nanobodies would be equally efficient in a way in treating Crohn's disease and Parkinson's disease, since they would lower the kinase activity in both. What we haven't tested so far, and what we for sure should do, is whether they are as efficient in lowering the kinase activity of all the mutants. We know that the wild type, kinase activity of the wild type protein is reduced, and also of the G2019S mutant. That's the most common mutation in Parkinson's disease. So we know that they work on both of these proteins. We haven't tested all the mutants yet, whether our nanobodies still bind to these mutants as efficiently, for example, and would lower also the kinase activity there. but my guess would be that they would. I'm pretty sure, let's say, that they would.
Excellent. Thanks, Wim. Our next question asks, do nanobodies bind to human antibodies, such as Ig?
I don't think they bind to those antibodies by nature. I'm not sure what's the question exactly, or the person asking the question is exactly referring to. So as far as I know, they don't bind by nature to other antibodies. Of course, probably if you would immunize with those antibodies, and they would be immunogenic enough in a llama or a camel to elicit an immune system, you would probably find nanobodies that bind to IgGs and IgMs, at least if they are different enough from the self antibodies that are produced by the camel, of course, because they should really distinguish between self and non-self in order to avoid any autoimmune diseases. But if they are immunogenic enough, I'm pretty sure that you could elicit an immune response and get nanobodies also binding to these antibodies.
Excellent. Thanks, Wim. Our next question asks, can this technique be used in any other non-brain diseases?
Yes, there are many more examples of nanobodies that are being used in or being tested, at least in clinical trials for diseases that are non-neurological diseases, because that's, of course, way easier in a way, because you don't need to get your nanobodies to the brain. And if your target protein that, for example, in cancer, if that would be a receptor protein that is exposed on the outside of a cell, that would even make it more easy. And in fact, there's one nanobody now that is really in clinical use, and that's a nanobody that binds to the Van Willebrand factor and is used as a drug against a disease called thrombotic thrombocytopenic purpura, which is a kind of rare form of thrombosis, and that's a drug under the name Cablivy, trade name Cablivy, that is now on the market, and that's the first marketed therapeutic based on nanobodies.
Excellent. Thanks, Wim. Our next question starts off by saying that there's some data that blood-brain barrier is compromised in Parkinson's disease. Is it compromised enough for nanobodies to go through?
In very late stages, probably yes, but that's, of course, the big issue with Parkinson's disease. If you want to find a drug that really alters the progression of the disease and really is able to kind of stop the progression of the disease, and for that there is still no drug available, it's of course crucial that in the first place you be able to have a diagnostic which is able to diagnose the disease in a very early stage. And there are still not that many or no diagnostics really that can diagnose in a very early stage. But so ideally you want to start with your treatment before the blood-brain barrier is so heavily compromised, because that would already be a quite late stage of the disease where both motor and non-motor symptoms would already be quite pronounced and quite far progressed. So yes, it's possible, I think, but only in very late stages, and you ideally want to start your treatment earlier.
Excellent. Thanks, Quinn. Our next one, I'm not entirely sure whether I quite understand this one, but hopefully you do. Wim, it's basically asking, can nanobodies develop antibodies in humans?
Oh, yes, I do understand, I think. I think the question is whether they are immunogenic or not. Well, in general, by nature, apparently llamas and camels are already a little bit more human-like compared to mice, for example. So by nature, they already elicit less of an immune response than the normal antibodies that would be raised in mice, for example. Still, it is kind of advised, and I think most companies, so also Cablivy, which is now on the market as a FDA and EMA approved drug, have been gone through rounds of humanization. So there are certain mutations, and they are quite well known, and it's quite standard procedure as far as I know. A number of mutations you can make on the surface of the nanobody outside of the really paratope where it binds to its target protein, that you can mutate to its human counterpart, and that makes it even less immunogenic. And especially for long-term treatments like you would need in very progressive diseases and chronic diseases like Parkinson's disease, it might be advantageous indeed to go through a humanization stage before really be using it in humans, let's say.
Excellent. Thanks, Wim. Our next question asks, so what are the advantages and disadvantages of nanobodies compared to classical antibodies?
Well, I already dwelled a little bit on that during my introduction. So one of the big advantages is that they are single domain. So that means that they are extremely easy to clone. You just need, in fact, one PCR to clone your nanobody. You can very easily engineer them. So as I already said, you can put any other type of protein at the C-terminus of the nanobody, fluorescent proteins, but also proteins that target towards the proteasome, signal peptides to target to specific organelles, and so on. So that's already one big advantage. They are very easy to produce and very cheap to produce. Let's say you can just produce them in E. coli, so you don't need any fancy hybridoma or whatever production systems. They are less immunogenic, as I already said, which can also be an advantage. They are very stable and robust. So you can leave them for 2 weeks at 37 degrees and they still didn't lose any of their binding capacity. And they are also more resistant to oxidizing conditions compared to classical antibodies. So non-reducing environment, I mean. So when you bring a normal antibody in a reducing environment, such as the cytoplasm of the cell, so you would reduce all your disulfide bonds and probably the antibody would not be functional anymore. You don't have that with the nanobodies, or much less. They have a disulfide bond, but they are less reliant on that disulfide bond, so you can more easily express them also in the cytoplasm of cells. And they are very small, which means that they can penetrate quite easily into different tissues and so on, which is immediately also one of the disadvantages depending on your application. So they get cleared very quickly from the circulation via the kidneys. So for therapy, sometimes people try to make them larger again. For imaging, it's an advantage that they get cleared very quickly, but for therapy, sometimes people want to make them bigger again. And maybe a last thing which might be important for people wanting to work with nanobodies, another what you could call disadvantage compared to classical antibodies. So the nanobodies only have 3 loops, like I explained, that protrude, while classical antibodies have a more flat surface. That, of course, makes these nanobodies extremely suitable to bind into cavities and to bind proteins in a conformation-specific way, but they don't like to bind to linear epitopes. So they do not bind to unfolded proteins, and for that, very often you cannot use them in Western blots, for example. So they do not detect the linear polypeptide chains after you unfolded a protein. So for that, you still might prefer to use classical antibodies.
Excellent. Thanks, Wim. I think we've got time for just one last question. And this one asks, have you tried to make a PROTAC from these nanobodies? And have you observed the synergy effect when you use 2 nanobodies simultaneously?
On both questions, I can say that we are planning to do that. We're still lacking hands to go fast enough to do every idea that we have in the time that you would, of course, ideally want to. But as I said, for example, the PROTAC strategy has been used in case of alpha-synuclein. So that's for sure a very exciting therapy, but it does not make use of the advantage of the nanobodies that we now have. So that would just, let's say, shut down the protein. While the advantage of the nanobodies that we are having now is that you might shut down certain aspects of the function of LRRK2 and not just completely, let's say, lower the levels of LRRK2. It might have other functions in the cell, so it might be advantageous to just, let's say, decrease kinase activity or even specifically RAP phosphorylation and not other phosphorylation of other substrates. And so, but we are thinking of that. And obviously, combining the 2 antibodies, that's something that we are doing as we speak. And we hope indeed to have a kind of multiplicatory effect of combining the nanobodies. Yes. Good point.
Thanks, Wim. Well, I'm afraid that's all we have time for today. I would now like to thank Dr. Wim Versées for his presentation and for answering your questions, and of course you, the audience, for taking the time to be with us again. Remember, you can watch this webcast again at any time on demand at nature.com webcasts. Thanks for watching, and I hope you'll join us again soon.
Have a nice day.